today's class, we'll be starting on the processes in recombinant DNA technology. And in the processes of recombinant DNA technology, primarily we need to exactly address ourselves to two important situations. That is, how do you separate the DNA from the cell? Initially, for genetic engineering or recombinant DNA to occur, we need to extract the sort of a DNA from the cell. As you understand, DNA is present in the chromosomes. Chromosomes are present in plant cells and it's in the region of the nucleus. So, it's very, very important that when we want to extract the plant DNA, we have to extract it by a biochemical process so that we get the pure DNA. And once we get the pure DNA need to be cleaved by means of restriction enzymes. Once you cleave it by means of restriction enzymes, which I learnt in the last class, we need to take the sort of a DNA and we need to separate or identify the gene of interest from the sort of a broken pieces of the DNA. So, we have two important techniques here which you are learning. One is with relation to separation of the DNA and next is the identification of the gene of interest. We will be talking initially about the separation of the DNA. Now, whether we take a bacterial cell or a plant cell or a microbial cell, the cell should be subjected to lot of digestion, because we need to break down the cell walls, we need to break down the plasma membrane, we need to break the different sort of an organelles, and later on we go to get into the sort of a method of how do you extract the sort of a free DNA, which is just like a spool of DNA or which is just like the sort of a noodle. So, when we use lysozymes, lysozyme is a sort of an enzyme which can primarily break down, especially it can cause lysis of the plant cell walls. So, when you take a plant or a microbial cell, if you add lysozymes, it leads to breaking of the sort of a cell walls. Now, this one again, when you use cellulase, it helps in breaking down plant cells, because plant cells are rich in cellulase. And similarly, when you take fungal cells, to fungal cells, when we add chitinase, the chitinase cell wall gets dissolved. Thereby, by using either a lysozyme or a cellulase or a chitinase, we can very easily break down bacterial cells, plant cells and microbial cells, and primarily with relation to all these by use of these particular enzymes. So, this is the sort of a first sort of a treatment wherein you take the cell and you add different sort of enzymes to break down the sort of a cell wall. So, this your objective is digestion of the cell wall. We are digesting the sort of a cell wall. Initially we had an intact cell wall, and if you want to extract the DNA, we need to break the cell wall. Now, after we break down the cell wall, we have got the different components of the cell organelles. The different components of the cell organelles need to be washed. So, thereby we add several sort of detergents. In the cold, under ice, we add several sort of detergents to the sort of remnants of the cell. So, when we add detergents, primarily it dissolves the sort of a lipid membrane. The lipids, which are an important component of the plasma membrane, are removed. You remove the sort of a lipid components, and later on, what is present within us, we have the DNA, we have the RNA and the proteins. In order that we eliminate the sort of an RNA, we need to add an enzyme called RNase. So, RNase helps in removal of the sort of an RNA. When you add RNase are removed. And similarly, when we add proteases, when proteases are added to the cell extracts, the proteins are removed. So, adding of RNase and protease will remove the sort of an RNase and protease will remove the sort of an RNase and the proteins, and all other molecules are again removed. Other molecules are removed when you subject the entire cell to chilled ethanol. Addition of chilled ethanol enables removal of all other molecules. Thereby, we understand that we come into the situation of isolation of the sort of a DNA. For the isolation of the DNA, it requires different cells. That is, first of all, in sequence we have lysosomes for bacteria, cellulase for plant cells, chitinase for fungal cells, use of detergents for other cell components, addition of RNAs to remove the sort of an RNA, then addition of proteases to remove the proteins, and chilled ethanol will primarily enable the sort of a DNA to come in the form of a sort of a churn. So, your DNA comes in a sort of a test tube, it will just go to form a sort of a spool like structure. It will go to form a sort of a spool like structure, which can just be removed, just like how you see a piece of a little bit of jaggery juice. So, this is the sort of a purified DNA. It is the purified DNA, and the purified DNA will tend to precipitate. Now, this is the first step towards the separation of the DNA. Next, we have a technique which is termed as the sort of a gel electrophoresis. The gel electrophoresis, or it is also commonly called as polyacrylamide gel electrophoresis. By using polyacrylamide gel electrophoresis, it is very easy to identify your sort of a gene of interest. Now, prior to the use of gel electrophoresis, we understand that we had at step number 1, we had isolated the sort of a DNA. Once we have isolated the sort of a DNA, the DNA is cut by means of restriction enzymes. Same is the case even with relation to the sort of a plasmid. Remember, in recombinant DNA technology, we require the sort of a source DNA. At the same time, we also have the sort of a plasmid DNA, wherein we have the sort of a plasmid vectors, and the plasmid vectors DNA also need to be cut. So, we use a common core of, or a common pool of restriction endonucleases. The common core of restriction endonucleases will cut the sort of a DNA of your sort of a host plant, as well as it will also cut the DNA of the cloning vector. That is, it will be cutting the sort of a plasmid DNA. So, once we cut the sort of a plasmid DNA, we have small pieces of genes on either side, and the small pieces of genes which are present on either side need be separated. So, for separating the sort of a genes, we require primarily a sort of a technique which is termed as gel electrophoresis. So, in gel electrophoresis, which is the next technique, it consists of the following features.